Friends, welcome. In this video I will discuss about the rest of the topics which we have not discussed yesterday. So let's get started. So as you can see that over here it is given reproduction. Now first of all we have to understand what do we mean by reproduction. Reproduction means when an organism produces one or more organism of its same kind, right? And also the ability of an organism to produce an offspring is the most basic features of life. Now the importance of the process of reproduction is for the continuity and survival of species. So for example over here, if I talk about reproduction, right? Under reproduction we have asexual reproduction. Now what is asexual reproduction? In asexual reproduction there is an involvement of only one parent cell without any fusion of gametes. So the offspring which is produced, since only one parent cell is involved over here, it is having exactly the same features as that of the parent. Now in sexual reproduction what happens, both male and female gametes takes place. That is, male we already know it is sperm, and female it is your eggs or ova. So when these fusion of gametes take place, the offspring which is produced, it is having the mixed characteristics of both the parents, and since both the genes are involved in him by carried by the chromosomes, it is not identical to either of the parent. Why? Because both of the features of both mother and father are involved. Now asexual reproduction basically occurs in lower forms of life, for example bacteria, fungi and all, and sexual reproduction occurs in very higher animals, for example if we talk about humans and all, lion, tiger, cat, dog. So asexual reproduction is again divided into many types. So over here, first of all I will discuss about budding. What is budding, right? What is budding? So budding, right? First of all, when I discuss about the process of budding, what is budding? In budding, basically what happens, a small bulb appears on the body of an organism, right? After some time what happens, this bulb which is there, it gets detached from the parent, develops into an individual and lead a simple life and lead its own life, right? So this was the process about budding. So for in this process what we have to remember, that first of all a small bulb is formed on the body of an organism. Then after that what happens, that then this bulb which is there, it gets detached. It means it gets separated from the parent, then it develops into a new organism and lead its life. So friends, this is human cycle. Like for example, you are only there. After some time, you will be separated from your parents, like for higher studies and all, and then you will lead your own life. Similarly, happens with these lower forms of organisms or any animal species or human animal species you take, right? After some time, the children have to be get separated from their parents to lead their own independent life. Just the process of reproduction is different. Now this diagram, can you see over here, this one, this is of binary fission. Now binary means two and fission means splitting. Actually binary fission is the simple division of a unicellular organism into two daughter cells. For example, this is a unicellular organism, it will divide into two daughter cells. both are roughly of equal size, then they will grow into an adult individual and again divide. It basically takes place in your like bacteria, euglena and all. Now then the next topic is budding is done, multiple fission. Now this is the diagram of budding you can see over here. This bulb appears. This bulb appears over here. Right. Now what is multiple fission? Friends, sometimes what happens, some organisms develop a thick protected layer around them. This thick protected layer is known as cyst. Now inside this cyst, cells repeatedly divide, means they divide and they form many daughter cells. Now when this cyst get overfilled and during favorable conditions it bursts, when it bursts the daughter cells which are there they get released and develop into a new organism. So the advantage of multiple fission is that the simple division of a single parent cell is there into many daughter cells. That is why the word multiple had occurred, and fission means split and means multiple splitting occurs. It basically takes in unicellular algae like your Chlorella and Chlamydomonas. So till over here I think that you all have understood. Now let me go next. Wait a minute. Fine. Fragmentation. Fragmentation, if talk about, so what is the process of fragmentation? In fragmentation, for example, there is a parent cell, right? If there is a parent, it will get divided into fragments, and these fragments will develop into a new adult individual, right? These fragments will develop into a new adult individual. Now, for example, these are two filaments. These are two fragments. Over here, a new filament is formed, and this will also combine, so they will together form a new individual. Now, spore formation. Spores formation basically occur in like bread mold, rhizopus, and all. Spores are basically tiny spherical unicellular bodies protected by a thick wall. Tiny spherical unicellular bodies protected by a thick wall. They are formed inside spore sac. Over here you can see that they are formed inside spore sac. So what happens during favorable conditions? Spore sac bursts, right? Sorry, spore sac never bursts. During favorable conditions, from the spores, many organisms are released on being released from the spore sac. Together, I want to say that during favorable conditions, many new organisms get released from the spore sac and develop into an individual. So, this process is known as spore formation. Let me go to the next frame. Now we will discuss about a new topic, vegetative propagation. Now friends, sometimes what happens if I talk about vegetative propagation, so some plants are there which are not able to grow directly from seeds, right? They don't follow this cycle. It's from seeds into a sapling then to a tree. They are grown from the vegetative parts of the plants like your stem, then your roots, leaves. So this process is known as vegetative propagation. It is asexual because no fusion of gametes take place. It can occur both naturally and artificially. First, I will discuss about naturally. By underground stem tuber. Now, tuber, if I talk about, so this is a basically swollen fleshy underground stem. It is a swollen fleshy underground stem, and it is having many buds known as the tuber is having many buds known as eyes, and when they are planted in the soil they grow into a new plant. Example of tuber is very famous. For example, potato tuber, right? Rhizome. This is a short branched underground stem which has distinct and internodes. These distinct and internodes bear buds. When they are soaked in the soil, they grow into a new plant. Example of rhizome you can take, for example, a ginger, banana, and canna. Corn, friends, if I talk about, so corn is basically a thickened round underground stem with swollen base surrounded by membranous scale leaves. They also have one or more buds planted in the soil, grow into a new plant. Example of corn is gladius, means your chukundar in Hindi we say it, and its English name is or its biological name is gladius. Bulb, if I talk about, so bulb is it is consists of an underground stem with a bud which soaked in the soil so grows into a new organism, new plant. Examples of bud are here, for example, onion, lily, and tulip. Biaxial stem in some plants such as strawberry, a slender branch arises from the base of the stem and come bends to touch the soil. It develops roots and buds at the point of contact with the soil. When it is planted in the soil, it grows into a new plant. And in some grasses like and mint and all, the stem grows along the soil, forming the roots at interval. When the plant is mature enough, it get separated from the parent and to lead an independent life. By roots, if I talk about in some plants such as your Canache and all, it has tuberous roots with adventitious buds. These adventitious buds, which are soaked in the soil, they grow into a new plant. By leaves and by leaves, if I talk about, so in some plants like a bryophyllum and all, it also basically consists of adventitious buds. And when the leaf falls in moist soil, this bud which is there, it develops into small plantlets. Then the leaf supplies food, the fleshy leaf supplies food and water to the plantlets. The plantlets grow on separation from the parent to lead an independent life. Now, if I talk about artificial methods, so vegetative proper now these processes takes a lot of time because they are natural. In artificial methods, this like such type of time is not taken because it is done by human. Cutting, so a stem is taken with an auxiliary bud, right? With the bud which is there at the lower end, it will develop roots, upper leaves. When leaves are planted in the soil, they grow into a new plant. Example of this you can take your sugar cane, your pineapple, fine bougainvillea. Now let me go to the next frame. Layering. In layering, what happened that a branch of the plant is taken and a part of it is covered in the soil. When the part which is under the soil developed roots, it is cut from the main plant, soaked in the soil, and developed into a new plant. Grafting. In grafting, what happens, two different varieties of a plant is taken, parts are taken. For example, if I talk about stalk and cycan, right? Stalk is the rooted stem, cycan is the stem cutting. Stalk supplies essential nutrients to the cycan. When they are tied together, cell division occurs and they give rise to a new plant. Grafting you can see in mangoes and all. For example, in mangoes you must have seen some mangoes are purple, pinkish, or combined color. That is basically done by grafting only. And if I talk about rose, so in rose also you must have seen various roses are of different colors, beautiful colors. If I talk about, friends, last method is tissue culture. It is also known as micro propagation, and this method is basically performed in the laboratory conditions. A small piece is cut from the growing tip of a plant, then it is growing in nutrient medium, and this part which is there, it grows into an unorganized mass of cells called callus, right? It grows into an unorganized mass of cells called callus. Then this callus is grown into a nutrient hormone medium to induce cell differentiation and formation of plantlets. Then when it is soaked in the soil, so it develops. So when it is soaked in the soil, when it is soaked in the soil, it develops into a new plant. Since this process is faster, it can occur in ornamental plants. Now, what are ornamental plants? Ornamental plants are those plants which are very brightly colored. For example, chrysanthemum, asparagus. So now at the last, advantages and disadvantages. If I talk about the advantages of vegetative propagation, so the basic advantages of the vegetative propagation is that the basic advantage of vegetative propagation is that first of all, it is used to grow seedless plants like a grapes, banana and grapes. Then the plants which are grown through vegetative propagation bear flowers and those by seeds. And further generations of plants which are grown through vegetative propagation have the same qualities as that of the parent. Disadvantages, since all are of the same variety, so if one is prone to infection can destroy the entire crop. Similarly, the further generation of plants which are grown through vegetative propagation can lose their vigor, and at the last there can be overcrowding because there is no dispersion. So with this friends, I end my video. This chapter is very important, so you should study this twice and thrice. Thank you, Jai Jagat. Have a nice day.